First of all, thank you, Dr. Tengku, for the kind and lively introduction. Okay. Of course, thank you to Deputy Vice Chancellor Office of Research and Innovation for organizing these sessions, which is a good platform for us to exchange knowledge and share our research experience so that it can promote research collaboration in the future. Okay, so without further delay, I will start my presentation. The title of my presentation today is From Microbes to Microbiomes. This presentation was meant to share with all what has been done and what is currently being done, and I promise, in the field of microbiology. Without further delay, let's proceed to the next. Ladies and gentlemen, I would like you to meet Anthony van Leeuwenhoek. Okay, he is basically the father of microscopy, where he is the first person to observe and describe microorganisms accurately using a microscope that he built by himself. Okay, at that time he didn't call microbes as microbe, rather he used the term animalcules or little animals. What we have here is an example of tree of life in biology. Biology scientists use tree of life to describe the diversity of organisms that inhabit the earth. As we can see here, under this tree of life, the organisms that inhabit the earth can be divided into three main branches: the bacteria, the archaea, and eukaryotes. By the way, humans, along with plants and animals and certain group of fungi, we are classified under the subdomain of opisthocont, okay, under the domain of eukaryotes. But what I want all to see here is that the diversity among the microorganisms is a lot more higher when compared to the diversity among eukaryotes, okay, which shows that most of inhabitants of Earth are actually the microorganism, not the macroorganism. So what are microbes? Microbes are actually any organism with the size of less than 10 micron, which explains why we need microscope in order to see them. So most of them are actually unicellular. Unicellular here means that that particular organism only have one cell that makes up his entire body. Okay, so bacteria basically consists only of one cell, unlike us humans. A single human can consist of up to 10 trillions of cells. Okay, that's how big difference between unicellular and multicellular. Microbes can be further divided into few groups, which is the protozoa or amoeba, the fungi, the archaea, the bacteria, and the virus. They are further divided based on either size or the cellular components or structure. For example, for protozoa here, they are the largest among these four groups. Okay. In fact, in terms of cellular components and structure, they are the most complex. As for archaea and bacteria, when compared to the protozoa, they are significantly smaller. And in terms of cellular structure, they are much more simpler, where you can find them in form of rods, which we call them bacilli, in form of sphericals, which we call them cocci, and some of them take the form of spiral, where we call them spirosheets. Last but not least, the viruses. So the viruses are the simplest form of microbes. Okay, they only consist. They are made of on. They only consist of a strand of either DNA or RNA coated with some proteins. Okay, so they only consist of two things: DNA or RNA and proteins coating. All right, next, why do we study microbes? Why do we need to know these little animals? The simplest answer is they can cause diseases. This is the main reason why we study microbes. Okay. The second reason, which not everyone know, is they affect our life more than we know. So how they affect our life, I will talk more about it in the later part of this presentation. So please stay tuned. Okay, let's go to the first reason, which we study microbe because they cause diseases. So the idea of microbe cause diseases is described in the germ theory. So the germ theory is not new. It has been established centuries ago, as early as in the time of Ibn Sina, which is around 1025. However, it takes around 800 years for this theory to be proven as valid. Okay, so basically the germ theory is proven by Louis Pasteur somewhere between 1860 to 1864 using this experiment that is designed by him, right? So basically the germ theory describes that disease is caused by the invasion of microorganism into the body, which is what Louis Pasteur tried to emulate here. So Louis Pasteur managed to prove that disease is actually can be caused by microorganism. So the next question is, which microorganism? As I mentioned before, the diversity among microorganisms is much more higher compared to the macroorganism. What I mean is that there are about thousands or millions of species of or strains of microorganism. So which one that cause diseases? How do we identify them? So here are some of the ways. There are two main ways here, using the classic methods or genetic based method. So under the classic methods, from the biological sample, it can be anything: urine, blood, food samples, swabs, anything you can think of that you think contain the bacteria that cause diseases. Then we inoculate them into bacterial culture media in order to produce more colonies of the bacteria. Once we obtain the colonies of bacteria, they were subjected for series of biochemical testing in order to identify the bacteria that we managed to grow from the samples. Okay. The next method is by using their DNA, which is the genetic based method. Why DNA? Because because it is generally accepted that every single organism contains unique DNA. So that's why genetic-based microbiology use DNA in order to identify the pathogen that cause diseases. So the workflow is almost the same. From the sample, you can either culture it first to produce more of the bacteria, or you can directly use the sample to isolate the DNA. So from the isolated DNA, we sequence the DNA, and then from bioinformatic analysis, we can identify the bacteria, and then after that we can devise a plan on how to cure the disease caused by the bacteria. One thing I would like to add is that at iPromise, we are more specialized in genetic-based microbiology, which is termed as microbiogenomics. So the core components of microbiogenomics is NGS, or any genotyping array of panels, bioinformatics, and databases. So actually, NGS is how the DNA of the microbes got sequenced, and bioinformatics and database is being used to read the DNA sequence. Why? Because in nature, DNA is not human readable. Okay, you need the assistance from bioinformatics and databases in order to read the DNA sequence. Here is actually what happened when we perform a DNA sequence project. So as I mentioned before, from the bacterial culture or the sample, we extract the genomic DNA. Okay, so from the genomic DNA, first we have to shear it. Depends on what technology that you use. Okay, for ours, we shear it into pieces. Okay, and then from these pieces of DNA fragments, we amplify it. Okay, we amplify in order to produce millions copy of each of these DNA fragments, which we call it library. Okay, so from the produced library, we subject it for sequencing. Okay, so basically we have a machine that read the sequence from the amplified library. Okay, from the reads which of the DNA sequence, as I mentioned before, you need bioinformatics tools in order to read it. So we use bioinformatics tool to read every single sequence that come from this fragment, and we need to reassemble it back into its original form. Okay, this one we must use bioinformatic tools. Okay, all right. Now we know how to identify microbes, and we also know how the genome is sequenced. All right. The next question is what to do with the genome that we have sequenced. Okay. Here are some examples of what scientists can do with a genome sequence. There are plenty here. So each of them serve different purpose. For us at iPromise, I guess we have done almost everything here, but in terms of microbiogenomics, we are more focused into specific detection of specific sequence for strain detection. We study genomes to gain insight into growth requirements and also to gain insight into the virulence of the particular pathogens. Okay, let's have a look into one example on how microbiogenomics can be used to study bacteria and the disease that it causes. So here are one of our recently published articles on microbiogenomics. So here the subject is Mycobacterium tuberculosis complex, or MTBC. Okay, basically this study is a collaboration between iPromise and Makmal Kesihatan Awam MKAK Sungai Buloh. What happened in this study is that MKAK provided us with pre-tested and deactivated MTBC isolates. After that, from the MTBC isolate, we extract the genomic DNA. And then from the genomic DNA, we perform whole genome sequencing, followed by bioinformatic analysis to study these two components of the MTBC genomes. Okay, polytyping and phylogenetic analysis, detection of variants associated with the drug resistance and virulence. Okay, here are some results from the published articles. As I mentioned before, we performed two types of bioinformatic analysis. The first one is polytyping and phylogenetic analysis, where we do it because in order to determine the lineages of the MTBC isolates. In the study of tuberculosis, it is very important for us to know the lineages of the MTBC that cause the tuberculosis. This is because certain lineages of MTBC are more prone to develop resistance towards the anti-TB drugs. For example, here in this study, we found that MTBC that comes from the Indo-Ocean or Beijing lineages are more prone to become resistant to anti-TB drugs like isoniazid, rifampicin, and ethambutol. Next, our second bioinformatic analysis is to detect the variants that is associated with drug resistance. Okay, so the result from the bioinformatic analysis reveal up to 20 mutations that can be associated with drug resistance. Okay, which means that the bacteria become resistant to the anti-TB drug because of this mutation that we found. Another thing that I would like to highlight here is that we managed to complete the analysis roughly around 1 week. Okay, this is why I highlight this, because only through microbiogenomics that you are capable of complete such analysis within 7 days. Okay, under the classic methods of microbiology, it's not possible to obtain such result within 7 days, because growing the mycobacterium to the MTBC inside the plate itself take at least 2 weeks. Okay, so from the after 2 weeks, you need to perform another biochemical testing, which can also take at least 2 weeks. So that is already a month. So that is sort of the difference of what you can get using the classical microbiological method and also the using the microbiogenomics. And I promise we also use microbiogenomics to study emerging infectious diseases. Okay, emerging infectious diseases is diseases that appear within a population or those whose incidence or geographic range is rapidly increasing or threatens to increase in the near future. So this emerging infectious disease can also is caused by previously undetected or unknown pathogen. Example of emerging infectious diseases is tuberculosis, influenza, SARS, MERS, and COVID-19. Another point that I would like to add here is that certain emerging infectious diseases like SARS, MERS, and COVID-19 are best studied with microbiogenomics. This is because this tree is actually caused by coronavirus. So since the pathogen of each diseases is actually come from roughly the same species, conventional biochemical testing might not be able to differentiate between them. It is only through microbiogenomics that we can differentiate between the pathogen of SARS, MERS, and COVID-19. All right, here are some updates on the microbiogenomics of COVID-19. So as of July 2020, there are about 54,000 of SARS-CoV-2 genomes have been sequenced worldwide, where 37 of them were isolated from patients in Malaysia. Okay, so the 27 of them is deposited by IMR, MKAK, UM, and IIUM. Okay. As for UITM participation in the COVID-19 microbiogenomics, at the moment, I promise is working together with Faculty of Medicine, UITM, where the Faculty of Medicine provide us with plenty of samples for us to sequence. Okay, then after the sequence, we will do the necessary bioinformatic analysis in order to study the mutation that might be unique in COVID-19 that isolated in Malaysia compared to the worldwide. Because at the moment, we believe that it was due to certain mutation inside the COVID-19 genome that leads to the aggressiveness of the pandemic. As you notice, in Europe or in US, the strain of COVID over there are much more aggressive in terms of spread, okay, compared to us, Malaysia. All right. So it might be due to the way we handle the pandemics, which is might be better than those US or Europe, or there are certain mutation happen in the Malaysian strain that leads it to become less aggressive in terms of spread compared to the Europe and the US strain. Who knows? That's why we need to study it. Okay, so let us finish the study first. All right, this one is a bit an offshoot because rather than studying bacteria that cause diseases. Here we are studying bacteria that didn't cause diseases. In fact, we rarely encounter this bacteria, which is Geovascular stimulators. This bacteria is actually an extremophile, or to be more specific, a thermophile, where this bacteria can live at temperature between 60 to 70 degrees. So actually, this bacteria was isolated from a local hot spring, which is I think Sungai Selayang hot spring. Okay, so why we study this bacteria? Because of its unique traits, because this bacteria actually can survive in environments that are hostile to life, which is this temperature. Okay, for the record, no microorganism can survive this temperature. Okay, this bacteria not only survive, but it thrive living in these conditions. So we want to know how did the bacteria do it. Okay, so this study involves 3 omics study. Okay, genomics, probiomics, and metabolomics. Okay, so what do we found from the study is that we managed to found about 60 proteins that is being expressed in the bacteria when it was grown at 70 degree, or when it was put under thermal stress. And then among those 60, there are 21, these are the 21 of the 60 that we predict to give the bacteria the thermophilicity trait. Basically, these are the protein that we think helps the bacteria to survive living at high temperature. Okay, so some of you may ask, so why bother needs to know about this protein? What is the use of this protein? Okay, this protein might have no use in real life, but what we want to know exactly, how does this protein can function in high temperature, while the same protein that exists in other organisms cannot. Okay, for example, certain protein here, okay, certain proteins here are also available in other bacteria. Okay, so by studying the factor that allow this protein to survive high temperature, we can sort of copy the factor and transfer it to other protein of interest and to make it more thermostable. Okay, for your information, thermostability is one of the desired character in industrial proteins. Okay, so industry really love proteins that have high that can that have high that are highly thermostable, because the industrial, most of the time industrial reaction, it occurs at high temperature, so that's why they prefer the protein to be high temperature for them to use the protein inside the to catalyze the industrial reaction. Okay, next we go to the microbiome study. Okay, previously most of my presentation are on microbes, which is we are focusing on a single microbes at one time. But for microbiome, we are at one time we are not studying one microbes. In fact, we are studying the whole community of microbes. Okay, so microbiome is actually a collection of microbes in a particular environment. Okay, and they often involve the genetics of the microbe as well. Okay, why genetics? Okay, so without genetics, actually you cannot perform microbiome study. This is due to not all microbes are viable. Okay, most species of microbes is actually cannot be cultured or cannot be grown inside a lab. Okay, so that's why the only way for us to notice their presence is through their genetics. Okay, how we how we detect their genetics is, as I mentioned before, in microbiogenomics, you don't have to culture the bacteria first. Instead, you can isolate the DNA directly from the sample. Okay, and then you can sequence the whole DNA that available inside the sample. Okay, so that's how we can trace the presence of microbes that is unviable. Okay, so example of actively studied microbiomes. Okay, it depends on what field. It depends on what field that the microbiome was interested in. For example, in climate changes, most scientists study the ocean microbiome, soil microbiome, and Arctic microbiome. As for us, who are more on health and well-being, we study gut microbiome the most, and some of us even study eye microbiome and mouth microbiome. But and I promise, at the moment, our focus is more on microbiome, gut microbiome. Okay. Remember earlier I told you that why we need to study microbes. First, because of they cause disease. Second, they affect our life more than we know. Okay. These are the example of how does the microbiome or microbes actually, community of microbes affect our life. Okay. So here you can see that they can even affect your dream. They can sort of affect your mental health. Okay. They also digest some of the food that we eat, okay, and a lot more, okay. Uh, as okay, here are some other interesting facts on human microbiomes. All right, I think the first one is quite interesting, as it says up to 90% of all diseases can be traced back in some way to the gut and health of the microbiome. I guess this is interesting because it is sort of in line with what our prophet said, which is most of the diseases comes from the stomach. Okay. And another interesting fact is that there are more than 10,000 species of microbes species that have been identified living in our body. Okay. So okay, here are some example of studies that have been done on human microbiome. So there are plenty of them, and most of them are quite recent also, which shows that this is the future of microbiology, perhaps. Okay. So in order to study the microbiomes, we still use microbial but there are specific terms that used to describe a study that study microbiome, which is metagenomics. Okay, so metagenomics is actually a tool for microbiome study, where for microbiogenomics we sequence, we perform sequence on one particular microbes. Okay, but in metagenomics, okay, we, as I mentioned before, we sequence the whole DNA that available inside the samples. Okay, so here is some a definition of metagenomics. Okay, so this one I think from Wikipedia: study of genetic material recovered directly from environmental samples. Okay, this one is a bit more specific. Application of modern genomic techniques to the study of communities of microbial organisms directly in their natural environment, bypassing the need for isolation and lab cultivation of individual species, just like I mentioned before. Okay, you don't need to culture it on a culture media, because most of the microbiome are not viable. Okay, then why is metagenomic? Because in order for you to study the effect of microbiome, you need to study all of them at once. Okay, because to a certain extent, microbiome, they works like organs. Okay, they work together to perform a particular function. Okay, so they are like organs. So if you study, if you didn't, if you study them separately, you will not get the true picture of what they have, what they have done, or what are the function that they carries. All right. And the second one is, of course, as I said, 99% of the microbial species are not viable, which means that you cannot culture them. The only way for you to notice their presence is through their genetics. Okay, there are two approaches in metagenomics. Okay, first one is called 16S metagenomics. The second one is shotgun metagenomics. Okay, the first one is it's more targeted metagenomic. The second one is that you, as I mentioned before, you sequence all available DNA fragments. Okay, the first one, since it was targeted, so the usage of it is quite limited, which is you can only study the composition of a particular bacterial population. And the shotgun one, since you cover all, you can study the composition and together with the function of a particular bacterial population. Okay, for the targeted metagenomic, which is 16S rRNA genes, okay, why the 16S is being targeted? Okay, why not other genes? Okay, so I think this one, almost every one of us have seen this. Okay, this is actually the central dogma of molecular biology, which describe on how life works at molecular level, where everything comes from the codes that encrypted in our DNA sequence. Okay, and then the DNA got translated into mRNA, and the mRNA got translated by ribosome into protein, where the protein is the one that carry the function. Okay. So from mRNA to protein, the translation of mRNA to protein must be done by ribosome because there are no other protein that can translate mRNA to protein other than ribosome. Okay, so that's why every single organism has ribosome. Okay, bacteria, archaea, protozoa, animals, plants, and human, they all got ribosome except for virus. Remember, virus only consists of DNA, or RNA string with protein coatings. Okay, so since all living organisms got ribosome, and then plus certain region of the ribosome are very specific to a particular organism, ribosome serve as biological ID for each of the organism. Okay, so if you sequence the ribosome genes, you actually have the biological ID that is specific to the particular organism. So using 16S rRNA gene, you can sort of know the identity of the microbes that present in your sample. So that's why we target 16S rRNA gene for the 16S metagenomic study. Okay, I promise we do both approach of metagenomics, which is the 16S metagenomics and the shotgun metagenomics. Okay, the example of study that I have here use shotgun metagenomics. Okay, so what happened in this study is that we have groups of rats. Okay, and then each groups will undergo diet intervention. Okay, which result in a groups of rats, which is the lean rats and the obese rats. Okay, and then the obese rats, some of them undergo probiotic intervention where we feed them with probiotics. For this diet intervention, we feed the obese rat with high fat diet to make them obese. Okay, so lean rat, we just feed them with normal diet. Okay, and after that, for the obese rat, we provide them with probiotics. Okay, and then to produce another groups of rats, which is obese rats on supplement. So for each of these groups of rats, we perform shotgun metagenomic analysis in order to see the effect of the diet onto each of these groups of rats. Okay, and here are the results. Okay, here we got obese rats, and these two are actually obese rats on supplement where they were fed by different types of probiotic products. Okay, and this one is the control. Okay, the lean rats. Okay, so here you can see that the population of gut microbiome is very much different. Okay, so let's say when you compare the obese with the lean, it seems like the one that contributed to the obesity, I mean the effect of high fat diet is it reduces the population of Bacteroides and increase the population of actinobacteria. Okay, so where we believe that because of this reduction and inclusion that leads to the obesity. Okay, as I mentioned before, from shotgun metagenomics, you can sort of know the composition, and also you can know the affected or effective functions by each microbiome population. Okay, this one is the composition, which means that what are the groups of the groups of gut microbiome that affected by the diet type that we provide the rats. This one is what are the function that is affected by the diet type that we provide to each of the rats, groups of rats. Okay, so here we can see that from the high fat diet that we provide, of course, as expected, the function the function that is affected much more related with fatty acid biosynthesis, triglycerol metabolism, and LPS metabolism. Okay, so here we can conclude that whatever we ate can actually beneficial to our microbiome, and at the same time can be bad for our microbiome. Okay, so our study here proves that. All right, last but not least, another study that I promised involving the gut microbiome. So this time we use the 16S metagenomic approach. So what happened in this study was we select students from Faculty of Health Science. Each of the students were asked to do the DAS21 test to assess their levels of stress, anxiety, and depression. Okay. After that, we collect their stools and saliva as samples for our 16S metagenomic analysis. Here are some results from the analysis. As I mentioned before, from 16S metagenomics data, we can only found the data on the composition of the studied microbiomes populations. Right. But it is not necessarily a bad thing, because from here we managed to identify groups of microbiomes that we suspected to be related or to induce either stress, anxiety, or depression. Okay. So what to do next is we can sort of proceed with an intervention study where we treat the students or patients who is under severe anxiety, stress, or depression with prebiotic or probiotics that capable of diminishing or minimizing the population of microbiome that we believe to induce stress, anxiety, and depression. As conclusion, microbiology is an ever-expanding field, as there's still lots for us to discover and learn. Certain areas like how they affect our life or how they got affected by our life are currently being actively studied by researchers all over the world, with the hope that in the future, there are possibilities for us humans to fully control, modulate, or even make the microbiome a pet. Okay, with that all said, thank you very much for your time. Thank you.
Thank you so much, Dr. Fakhruzzaman, for that insightful presentation. Now, I open the session with the question and answer. Those who have questions to ask our speaker, you feel free to unmute your microphone, or you can also type your questions in the chat box, and I will try to assist Dr. Fakhruzzaman to read your question. Any questions from the member of the floor? Okay, one question from Siti Sohaila. Doctor, have done any soil microbiome related to climate change?
Okay, at the moment, as I mentioned in this in my presentation before, for any microbiome study, we only focus on the health. Health-related issues. So at the moment, I haven't studied any soil microbiomes, except for the Geovascular. But the Geovascular is not really a microbiome because it is more like of focus on only on the Geovascular stimulators. Okay, so it is not really considered as soil microbiome.
Okay, another question from Asdayanti Muslim. May I know what kind of bioinfo analysis conducted on microbiome analysis, on micron and biome analysis?
Okay. Based from my experience here, along with colleagues that have done the microbiome analysis, okay, there are a few tools that are available. One of it, of course, the one that you already suggested, which is the Kimi. Okay, one of my colleagues also use Metagenomics. Okay, that one is sort of a fully automated tools for metagenomic analysis. The Kimi is actually not only fully automated. Okay, so for me, frankly, for beginners, I would suggest to. If you want, depends on what you want to gain. Okay, some of us we would just want we just want to go through the easy way, which we prefer a fully automated tools. So we just give the input, which is the data, and the tools will analyze for us and give us the proper report. But for me myself, I prefer something that is more semi-automated, so that you can learn more about the process or the bioinformatics analysis involved in conducting microbiome analysis. Okay, but beside the Kimi and the Metagenomics, I believe there are also other tools that are available out there. It's just that a matter of we look for it. Okay, I hope that's answered your question.
Okay, for Asdayanti, maybe you can try also MG Rasya. So another question from Asdayanti, do I promise consider to organize bioinfo workshop in the future?
Okay, that one is a good question actually. In fact, by right around this year, which is between July and September, we're supposed to have a bioinformatic workshops. Okay, but since we are at the moment still under PKPP, so all of our scheduled workshop has been postponed to some dates in the future. But of course, yeah, we do have plans to organize such seminar for bioinformatics. Anything else? Yeah.
Okay, question from Shamsul Bahrain. Any studies in iPromi study the relation between the intestinal microbiome and helminth parasite common in Malaysia?
At the moment, no. We didn't actually study the relationship between the gut microbiome and the parasites. So yeah, no.
All right, I think that's it for today. Okay, so thank you so much, Dr. Muhammad Fakhruzzaman. And also, those are still currently online with us now. Thank you so much for your attendance. And also thanks to the organizer. So just a quick reminder that we have another last session for the I Promise Fellow to present his work, which is on this coming Wednesday. So stay tuned, yeah? All right. So don't forget to fill in your attendance. And that's all, I guess. Thank you so much. Yeah, catch up later.